And welcome back to the summer term. So what we're going to be doing today is we're going to be revising cells. So you've already had a look at this on Doddle, but we're going to do a big mind map of cells, and then I'm going to be setting you some other tasks to do after you finish this mind map. Now, before we get into the mind map, I want to just explain to you how the course is going to go for the next 5-6 weeks till half term, and go through the little plan with you. So this is this first half of the summer term. So we're going to have 5 weeks during which we're going to review the content, all the content that you've done so far. So in this first week, we're going to be looking at cells and biological molecules, and then we're going to go on to do nucleic acids, enzymes, and so on and so on. So there's going to be 2 topics per week. And for each topic, I'm going to do 2 YouTube videos. Sorry, for each topic, I'm going to do 1 YouTube video, which is going to be a revision mind map. Now, that will be quite a lot of work. That will take you a good hour to do the mind map, and potentially longer if you want to bring in extra detail from your textbook and from Doddle presentations that I'm also going to set. Once you've finished that, I'm also going to set you some multiple choice questions, which, again, I'm going to do a video going through those multiple choice questions, and you're going to self-mark those. And then in addition, I may set an essay question once a week. So for each week, there's going to be a YouTube on the mind map, and then there's going to be a YouTube on the multiple choice questions going through them. So there's going to be 4 YouTube videos a week, in addition to the multiple choice questions that you're going to be doing and marking yourself. Okay, so that's the workload, and that's going to cover both my side and Ms. Parvez's side. So that's going to be your workload for each week. There might be a little bit more work this first week because I think Ms. Parvez is just finishing off the last little bit on plant transport. Okay, so let's get into the mind map for today. So first thing is you find this PowerPoint, which I'm going to set you on Teams, because I've set you this PowerPoint here with these pictures. So these pictures are a little bit bigger. So my suggestion to you is print those out on A4. If you've got a color print, that is amazing, but it doesn't have to be color. So print those out on A4, and then you can cut out the pictures, and your mind map is going to be A3. So if you don't have A3 paper, completely fine. 2 things you can do: either take 2 pieces of A4 paper and just tape them together down the middle, and then you've got an A3 blank piece of paper which you can do your mind map on. Or if you want to do this in your exercise book, then just open up a double page spread of your exercise book, and then you can stick these pictures in and do an A3 mind map. Okay, so get yourself ready, print this off, get the pictures ready, and once you've done that, you can stick them in, and you can either sort of stick them in as I go through it in an order that feels good to you, or you can stick them in the way that I've done, and then we can sort of just annotate. So this mind map is going to take about an hour, but you can pause the video and do it in sort of bite-sized pieces. Okay, so let's get going. All right, so first of all, I'm just going to go through the main parts of the mind map and show you each different little bit that we're going to be looking at. So over here, we're going to be looking at the scale of living things. Okay, so how big are cells and the parts of cells. Then over here, we're going to be looking at a eukaryotic animal cell, a plant cell. We're going to look at the nucleus and how the nuclear envelope and rough endoplasmic reticulum kind of link together. We're going to be looking at the stages in the production of a protein over here. Briefly going to look at 2 organelles, the mitochondrion and the chloroplast, in detail. We're going to be talking about magnification and sort of calculating the magnification on an electron micrograph over here. And then we're looking at prokaryotic cells down here and how they are different from eukaryotic cells. So we're going to start over here by looking at scale. So let's zoom in a little. Okay, so all these images are taken from your textbook. So if you want to find the images in your textbook and follow along, then that's no problem. All right, so what have we got here? This is an atom. Only about 0.1 nanometers. Then if we get a little bit larger, we've got 1 nanometer here. This is a phospholipid. And a phospholipid is around about 3.5 nanometers, roughly. Over here, we've got some organelles. So this is a ribosome, and this is a protein here, around about 10 nanometers. It's worth noting that the phospholipid bilayer equals 7 nanometers in width. That's the phospholipid bilayer, so two of those together. Okay, so getting larger, we're talking here about a virus. So this is a flu virus, around about 100 nanometers. By the way, coronavirus is about here, which is about a largish virus, and that's about 120 nanometers. Now we get to a micrometer. I'm just going to draw that to the side so you can really see it. 1 micrometer. So that's the Greek symbol mu, micrometer. Now, actually, I haven't defined these units. So let's take a step back and do that for a second. So what is a nanometer, a micrometer, a millimeter? Well, 1 nanometer here is 1 times 10 to the minus 9 meters. Okay, so that means it is 1 billionth of a meter. 1 micrometer over here is 1 times 10 to the minus 6 meters. So that means that is 1 millionth of a meter. A millimeter over here is 1 times 10 to the minus 3 meters, so 1,000th of a meter. And then a meter is a meter. So to go between these units, if I want to go from nanometers down to micrometers, there are 1,000 nanometers in a micrometer, and there are 1,000 micrometers in a nanometer. Okay, so let's go back to our scale. Here we have 3 organelles. We've got the mitochondria, the chloroplast, and this, actually, sorry, that's not an organelle, that is a bacterial cell. Okay, so all those are around about the same size. Later on, we're going to see that there's a relationship between these things, because mitochondria and chloroplasts, they both used to be prokaryotes, free-living prokaryotes, and they were taken in by larger cells, and they became sort of organelles within those larger cells, and that's one of the reasons they're all the same size. Okay, so now we're getting into sort of human cells. So these here could be human cheek cells or other types of human cells. And this here is a human egg cell, so a bit bigger. Human ovum. So human cells are around about 10 micrometers. Human egg cell is around about sort of this size, close to 100 micrometers. And that is just detectable with the human eye, just on the very, very borderline. Now we get to plant cells. Plant cells are a little bit bigger. So this would be an onion epidermis cell. And that would be about 200, up to 200 micrometers. This would be a very large and complicated eukaryotic cell, something like an amoeba, which are up to 1 millimeter, very, very large for a cell. And then this is a chicken egg. Okay, so chicken egg is somewhere around about, I don't know what, 5 centimeters, a bit larger maybe. So that's the scale that we're sort of working at. Now in biology we really don't go sort of beyond this kind of point really much. Okay, so beyond here is kind of the realms of chemistry and physics up here. But of course we do go larger than the 100 millimeters. We can obviously be studying animals as large as the blue whale, which I think is about 30 meters long. So we can be studying things that are larger. And of course, if we're not looking at cells, we can go even larger. We can look at ecosystem levels and sort of the relationships between organisms that live in a habitat that might be several hundred kilometers across. But for today's lesson, we're going to be focusing on cells. So that's the scale that we're going to be looking at. And now let's look at a eukaryotic cell for the next thing. Okay, I almost forgot to mention what these two bars down here are. So this shows how we can see cells, organelles, parts of cells using our eyes and using magnification. So first of all, the human eye can see things just about, if you've got really good vision, down to about 100 micrometers. That is 0.1 millimeters, but that's on a good day, and that's just the limit of our vision. If you want to look at something small, you've got to use a microscope. So two types of microscope that we use in biology. The first one shown by this red bar is the light microscope. Now, the light microscope has its advantages, but there are limitations to a light microscope. And one of the limitations is the maximum resolution. The maximum resolution is about 0.2 micrometers, okay? And that's using special techniques, but practically in the lab, in a school lab, about 1 micron is about 1 micrometer is the finest level of detail that you can see. And if you have two objects that are closer than about a micron in a school laboratory, they will appear as a blurry blob that is one object. You won't be able to tell the fact that they're two objects, and that's what resolution means. So if we now look at this bar over here with an electron microscope, so the electron microscope has uses electrons instead of visible light, and this has a much greater resolution than the light microscope, and actually its maximum resolution is around about 0.1 nanometers. And again, scientists are kind of pushing the limits of this all the time, but that's the ultimate resolution that we can get. So because of that, we can study things at greater detail, and it's because of the greater resolution that we can zoom in more. You could zoom in more with a light microscope. Yes, you could. You could magnify things as much as you wanted to, really, but it would just be blurry, and you wouldn't be able to see the detail. So it's really the resolution that's key. You can add little extra notes about the benefits and the drawbacks of these two different microscopes. For example, light microscopes, you can look at living and moving cells, whereas in electron microscopes, the specimen has to be dead and specially prepared. Okay, so now let's zoom in, and now let's start to look at for example, a human cell. So this here is a typical human cell. We're going to look at the different parts of that cell. Okay, so first let's look at the center of the cell, possibly the most important thing, the nucleus. So the nucleus is here, and it contains the genetic information of the cell, and inside the nucleus we've got the nucleolus. And then we also have, surrounding the nucleus, we have the nuclear envelope. So, nucleus, of course, contains DNA. And that DNA in eukaryotes is found as chromatin, which is DNA associated with histone proteins, and the DNA is kind of wound around those proteins to kind of pack it more tightly. So let's put that in as well. The nuclear envelope here is a double membrane. In fact, we can see that up here. We're going to look at this later on. This is the nuclear envelope here, and it contains these holes in it up here. So those are called nuclear pores. I'll actually just label one over here. That there is a nuclear pore. Okay, so moving around, what other organelles do we find? Well, we've got mitochondria here, and that is the site of aerobic respiration. We've got to say aerobic respiration there. And that's because there's anaerobic respiration that happens outside of the mitochondria. So the mitochondria is the site of aerobic respiration. And over here, we can see this membrane structure. Now that's actually linked right into the nuclear envelope. We've got two types of this membrane structure here. We've got the rough endoplasmic reticulum, and then down here we've got the smooth endoplasmic reticulum. Okay, so the rough endoplasmic and the smooth endoplasmic reticulum, why are they different? Okay, so the rough endoplasmic reticulum it contains embedded ribosomes, and therefore it's the place where proteins are synthesized, and especially if these proteins are going to be packaged and delivered outside of the cell, then the proteins are often synthesized on the rough ER. And then we're going to look at that more in this diagram over here as well. Whereas the smooth endoplasmic reticulum is the site of lipid and steroid synthesis mainly. Okay, what else have we got? Over here we've got the Golgi apparatus, one of my favorite organelles. And it's kind of like a postal delivery depot. It receives vesicles and proteins, it modifies them, and then it sends them to where they need to go. And if you want to go deeper into how that actually works, slightly beyond the syllabus here, what happens is vesicles arrive at this face of the Golgi over here, which is called the cis face, and then these vesicles kind of merge and they move through the Golgi, kind of through these layers of these membrane sort of sacs of the Golgi, and then when they exit over here, they've kind of had their modifications added. So they may have had carbohydrate groups added to them, or even lipid groups to produce lipoproteins or glycoproteins. And then they reach this trans face of the Golgi, and that's where they're sort of sent to wherever they need to be. For example, they may be sent outside of the cell, they may be sent to the mitochondria, or they may be sent to a different organelle, wherever it needs to be. And then finally, we've got two extra things. We've got over here, we've got a lysosome. And down here, we've got this is the cell membrane, but when we speak about it in terms of A level, we call it the plasma membrane or the cell surface membrane. You can use either of those two terms, so I'll call it the plasma membrane here, plasma membrane, or the cell surface membrane. And the reason we call it that is because there's loads of membranes in cells. So if you just say the cell membrane, that doesn't tell you which cell membrane, because there's lots of different ones. So you have to say the cell surface membrane or the plasma membrane there. Now, we didn't talk about what the lysosome does, so let's quickly talk about that. So the lysosome contains digestive enzymes. So these could have two roles. They could be, if this cell was a white blood cell, then the lysosomes would be responsible for digesting any bacteria that are taken in by phagocytosis or breaking down any material that's been engulfed. Whereas in non-white blood cells, lysosomes are also involved in what's called autolysis. So that means breaking down stuff in the cell that's just worn out and it's just a bit old and knackered, and the enzymes will kind of digest old defunct proteins back into amino acids so that new proteins can be synthesized. So autolysis or phagocytosis are two roles of the lysosome. Okay, so that's the major organelles of the eukaryotic cell. And now let's contrast that to a eukaryotic plant cell. That was an animal cell. Now let's have a look at the plant cell. Down we go to the plant cell. Okay, so the plant cell, first of all, has a lot of features that are also present in the animal cell. So first of all, let's go ahead and label those. Okay, so all those things are found in the animal cell as well. So we've got the Golgi apparatus, we've got the mitochondrion over here, smooth ER, rough ER, nucleus, nucleolus, nuclear envelope, and ribosomes. I don't think I actually labeled the ribosomes above, so let's quickly look at that. Yeah, here are the ribosomes in the animal cell studded on to that rough ER. Okay, so what about the things that the plant cell has that is different from the animal cell? Let's label those and actually let's highlight them in a different color. So let's label those now. Okay, so the things that we've added that aren't present in the animal cell, we've got the chloroplast here. That is the site of photosynthesis. We've got the amyloplast, which is it's another organelle similar to a chloroplast. They're both called plastid organelles, hence the plast bit at the end. But this basically just stores starch. We've got the vacuole, which is filled with cell sap. And that's actually really important in maintaining the shape and the structure of the cell because it regulates osmosis and basically water flows into that vacuole and kind of inflates it almost like an inner tube inside a inside a tire, and it kind of maintains the shape of the cell. And that vacuole can actually take up a very large proportion of the volume of the cell, kind of pushing everything else out to the sides. So that is responsible for turgor pressure to regulate the shape and size of the cell, the kind of pressure inside the cell. Then we have the cell wall, which is made of cellulose. And these are cellulose fibrils, we call them microfibrils, and they build up into something called macrofibrils. And those macrofibrils actually kind of cross-hatch like this. So the cell wall is kind of almost a cage formed of layers and layers of these fibers lying on top of each other. So it's important to recognize that cell wall is completely permeable to water. And that it is responsible for structural support. And what I mean by that is it resists that turgor pressure. So we have the vacuole sort of pushing out via the water pressure inside, and the cell wall kind of resisting that pressure. So plant cells will not burst if they're placed in pure water. They'll just swell up, whereas an animal cell will because it doesn't have that cell wall. Okay, so we've gone over the main organelles and features of the animal cell and the plant cell. Now we're going to look at a little bit more detail on just a few of the key organelles. So first of all, we're going to look at how the endoplasmic reticulum kind of are all connected, how the nuclear envelope sort of flows right into the rough ER and then the smooth ER. We're going to do that by looking at this area down here. Okay, so first of all, let's label the key features of this diagram. Okay, so that's the main features of the endoplasmic reticulum network sort of labeled. So I just wanted to point out that it's joined to the nucleus. So this is the nuclear envelope. And we can see that there's a sort of continuous flow from inside the nucleus here through these little nuclear pores into the cisternae, which form which are the sort of fluid-filled, membrane-bound spaces that make up the rough ER. So this first layer is rough because you find a lot of ribosomes on it, and as you move further out, you can still sort of go from one cisternae to the next through these little joins. Then you get to the smooth ER, which is where lipids and steroid synthesis occurs. If you'd like to, you can add more detail to this diagram here by using your textbook and putting some extra notes around. Now let's move over and talk about a different key organelle. Over here, I'm going to label the mitochondria. So let's first of all label the key structures. Okay, so here are the main features of the mitochondria. First of all, it is a membrane-bound organelle, and actually it is surrounded by two membranes. There's an outer membrane, and then there's an inner membrane underneath it. And between those two membranes, there's this intermembrane space. Now, the inner membrane is actually folded like more. So it kind of folds and forms these kind of sheets of membrane that sort of extend out into the middle of the mitochondria. And those are called cristae, or singular is crista. And then the actual kind of the liquid inside those two membranes is called the matrix. If you wanted to call it the sort of mitochondroplasm, that's kind of how to think about it, but we call it the matrix. So here is a electron microscope image of a mitochondria, and I'll try and highlight those features. So first of all, the outer membrane is sort of there. These sheets here are the cristae. The matrix is any of the kind of liquid space just about there is some matrix. Intermembrane space, pretty difficult to find, but it is just about there. You can just about see the gap between the two membranes there, and the inner membrane, of course, would just be underneath that. So you have to recognize how that image relates to this sort of 3D representation. Okay, we've got one more to look at, one more key organelle to look at, and down here we've got the chloroplast. Let's actually just first of all link this mitochondrion here, so we know that's what that is. and the chloroplast is here. Okay, so first the key features of the chloroplast. Okay, so when we look at the chloroplast, again, it is a double membrane-bound organelle. We've got an outer membrane and we've got an inner membrane, and then there's a small space, fluid-filled space, in between those two membranes. Inside of those two membranes, in the chloroplast, we have another system of some free sort of fluid space and then some membrane compartments. So inside we have the general kind of liquid inside the chloroplast we call the stroma. And then we have these stacks of membranes called thylakoids. And these thylakoids kind of stack on top of each other, a little bit like a tower of pancakes, and they form something called a granum. So this would be a granum here, this whole thing here. I'll just sort of draw around that. So that would be one granum there. And then between the granums, we have these little connecting bits called the intergranal lamellae. And in year 13, you'll learn a bit more about where, how all those different things relate and what happens where. But for now, we'll just look a little bit of detail. So stroma, that's actually the light independent CO2 fixing stage of photosynthesis. And on these thylakoids and the grana that are sort of built out of them, that's where the light dependent stage, which harnesses the energy from light energy and uses it, converts it into chemical energy using chlorophyll. So chlorophyll is involved in that process. Okay, so there's just a little bit of detail about how the chloroplast works. Also, I'm going to add a bit more about the mitochondria. So mitochondria So a bit more detail about the mitochondria there. So actually in the matrix, which is the fluid in the mitochondria, that's where something called the Krebs cycle happens. And it's basically taking a carbohydrate molecule and chopping it up into smaller and smaller pieces. And that actually produces carbon dioxide. And then it's these little cristae, the membrane bits, where something called oxidative phosphorylation happens. O2 is used up in that process, and actually that's where ATP is generated. Down here, let's also compare the structure of the chloroplast in 3D to this electron microscope image. Let's see if I can match them up. Zoom in a little bit more. Here we've got a stack of thylakoids. This sort of thing here would be a stack of thylakoids or a granum. An individual thylakoid would just be any one of these kind of pancake-looking things there. Intergranal lamellae, just about see. Ooh, kind of tricky. I suppose there is just about one, maybe about there. Joining two thylakoids. The stroma will just be here. Just label that there, stroma. And then we have the two membranes, a bit difficult to see in this diagram, surrounding, that would just be sort of around here. So you can see how this structure compares to this one here. Other things that you sometimes see in chloroplasts as well, which you might detect here, these black dots there, that could be a starch grain, which can act to store the carbohydrates produced in photosynthesis. And sometimes we also have lipid droplets. Difficult to tell what these little yellow dots are. They might be lipid droplets as well, potentially. Starch grain is maybe this one. I'll just label it there, SG, starch grain. We've looked at the scale, we've looked at a typical eukaryotic animal cell, we've looked at a typical eukaryotic plant cell, and we've looked at all the organelles within them. So this might be a good place to pause and to use your textbook to make sure you really understand the function of all those organelles. Our mind map's getting a bit crowded, but potentially you can add a bit of notes about the function of each of the organelles somewhere. And once you've done that and had a little bit of break, come back to the video. We're going to push on and look at how the organelles work to make proteins, electron microscope pictures of a eukaryotic cell, and also the difference between prokaryotic and eukaryotic cells. Okay, so pause the video, get that done, and then come back. Okay, so welcome back if you just had a short break. We're going to now look at this down here. So this is a bacterial cell or a prokaryotic cell. Now the first thing to notice about this is that it is a lot smaller. So remember, if we look back over here, the typical prokaryote is only about 1 micron compared to human cells that are about 10 microns. So if this was really to scale, this would be about 10 times smaller than this over here. Okay, let's zoom in and have a look at this prokaryote and label the features. So the first obvious thing about the prokaryote is that it doesn't have a nucleus. So that's actually what prokaryote means. Prokaryote comes from Greek, so karyo means nucleus and pro means before. So it means before the nucleus even evolved. So we do have something called a nucleoid, which is kind of where the DNA is. Okay, but it's not membrane bound. So actually the DNA is a loop of DNA, kind of in the middle, and it's just sort of free floating, and there are no histones associated with that DNA. So it's kind of sometimes called naked DNA. It's just floating about in the nucleus. And prokaryotes also have other bits of DNA as well. They also have plasmids, which is a smaller circular loop of DNA, which can carry other genes. Now, sometimes plasmids, not only, but often they carry genes. They might carry genes for antibiotic resistance, for example. And it's also worth noting that plasmids can be used as vectors for genetic modification, which you may have met at GCSE, and we'll go further into that in year 13. Okay, so I'm just going to go ahead and label all these other features, and we're going to talk about them. Okay, so those are the features of the prokaryotic cell labeled. Now let's look at a few of those. So there are many similarities between prokaryotic cells and other eukaryotic cells. For example, they've got cytoplasm, they've got ribosomes. Now the ribosomes actually are a little bit smaller than the eukaryotic ones. So they're smaller ribosomes, and we would say that they are sort of 70S ribosomes, and the S refers to a kind of size. It's actually to do with how quickly they separate down when you ultracentrifuge a sort of smashed mixture of cells. So they're slightly smaller 70S ribosomes. The eukaryotic ones, if you want to add that, are called 80S ribosomes. They're a little bit bigger, kind of gives you an idea of how much bigger. So the ribosomes are a bit smaller, cytoplasm the same. What else? Plasma membrane, much the same. Phospholipid bilayer. Cell wall, similar to a plant cell wall, except made of a different material. So it's made of peptidoglycan. And that's the more typical name. Sometimes it's also called murine. I'm not really sure how to pronounce that. And that's important. That peptidoglycan is a mixture of peptides and carbohydrates linked together. And it's an important molecule because we don't have it in our eukaryotic sort of animal cells. So often antibiotics target the synthesis of that peptidoglycan or interfere with the way that it links together, which cause the bacteria to kind of pop and explode if you get rid of their cell wall and they're still under that osmotic pressure. They just kind of pop. That's how penicillin works, for example. So I'm just going to add that down here. In fact, another antibiotic target in bacterial cells is actually the small ribosome as well. I think it's some of the tetracycline and things like that. I think those antibiotics, target the ribosome of the prokaryotes, which is slightly different to eukaryotic ribosomes. Okay, what else? The other thing that we should look at are these pili. Pili are pretty interesting. I'm just going to change colour here, why not? So pili are small sort of hair-like structures. And they can be involved in a few things. They can either be involved in kind of allowing multiple bacteria to kind of lock together in what's called a biofilm, so like a colony. And they can also be used to transfer genes from one bacteria to another. It's called horizontal gene transfer. So basically, if two bacteria sort of come close to each other, one pilus can kind of stick out and transfer genes into the other bacteria, which is one of the reasons that antibiotic resistance is a problem, because bacteria can kind of pass plasmids from one to the other, which might carry antibiotic resistance genes. And the other thing is they have a waxy protective cuticle around the outside. There are differences in types of bacteria. Later on, you might learn at university about gram-positive and gram-negative bacteria, and they have slightly different types of cuticle around the outside. And that's one of the reasons that some bacteria are more difficult to treat than others, because of this waxy protective cuticle that can protect them from medicines we might use against them. Okay, so that's the overview of the structures found in the prokaryote. Let's look at the main features that are different between prokaryotes and eukaryotes. Okay, we've got smaller ribosomes, we've got no nucleus, and the DNA is just sort of naked or without histone proteins. What else have we got? We've got with a small size, and also we have these flagella, another thing to add, which are slightly different to what we have in eukaryotes. So the flagella in prokaryotes are for movement, and they just spin like this from the base, like a kind of propeller. They spin round and round and round and round, and bacteria kind of power along with these as like little propellers, moving them through liquids. And eukaryotes have something a bit different. They have something called an undulipodium, or also a cilia, and they're a bit different because undulipodiums can kind of move all the way along, whereas the flagella spins from the base, and they have a slightly different structure. We didn't actually label an undulipodium on this eukaryotic cell, but potentially you could draw one in. Why don't we just draw one in now? It would kind of look a bit like this, sort of like that. And I think you may have already read about this in your textbook. You could add little notes about this undulipodium and why it's different from other flagellum. Okay, so that's the main differences. Those are the main differences between eukaryotes and prokaryotes. Okay, on we go to the next thing. Let's do a review activity now. So here we've got an electron micrograph. So that's an image taken with an electron microscope, and that shows plant cells. We looked at plant cells earlier, so hopefully you'll be able to look at this diagram, this picture, and identify what all these different organelles are. So perhaps you'd like to do that in pencil. Pause the video for a minute or two, see if you can label all these different letters, and then come back to this when you're done, and I'm going to label them with you. Okay, so the first two things you might have noticed up here, like it started, the cell wall is this kind of thicker layer, and the cell membrane is just underneath it there. So the cell membrane, remember, is underneath the cell wall. What's next? This large empty space here is the vacuole filled with cell sap. This one may be a little bit difficult to really decide which one it is, but I would say that that is a mitochondria. And really it all comes down to the pattern inside. You have to kind of try and look and see if you can see, is it cristae that you can see, or is it the thylakoids stacked like that you can see. This one here, I think H is a chloroplast. G there looks like the Golgi apparatus to me. The Golgi apparatus kind of looks a bit like a weird jellyfish. So if you see something that looks a bit like a jellyfish inside a cell, it's probably the Golgi. This whole thing from W to X, this is the nucleus. That there would be the nucleolus, and therefore the thing on the outside of the nucleus has got to be the nuclear envelope. And if you look really closely, you can almost see that there are two membranes there, not one. So that's the nuclear envelope. What else have we got? C. Gosh, I have no idea what C is, to be honest, but tiny little dots there. I'm going to say that's probably lysosomes. And you wouldn't really be able to see much else. It's strange that I can't see a chloroplast. Oh, sorry, there's the chloroplast. You wouldn't really be able to see much else there. You know, all these little dots, little blobs in the cell. Who knows what they are? They might be ribosomes, they might not, because the resolution is getting a bit. Those objects are getting a bit too small, really, to see with the resolution of the electron microscope, or just difficult to identify what's a protein, what's a ribosome. One more thing maybe we might want to label is this, the outer layer of the vacuole, that is called the tonoplast. Okay, so now let's look at almost the last thing, the penultimate thing. How do we use this formula triangle up here? Let's change color once again. I'm going to pick purple. Okay, so what do these letters stand for? So I is the image. A is the actual size in real life, not down a microscope, the actual size in terms of the cell, and M is the magnification. And magnification is normally written like times 100 or times 20,000 or something like that. Actual size, just a little note here, you need to remember that 1 micrometer is equal to, it's 1,000th of a millimeter, so that would be 0.001 millimeters. And then you also need to work with nanometers as well. So if you are working with nanometers, you might need to convert them into millimeters or micrometers. So remember, 1 nanometer is 1/1000th of a micrometer, or 1 billionth, or 10 to the minus 9 of a meter. Okay, so how about a practice calculation? So you might, for example, be given a magnification calculation, and let's say that someone measured this line from W to X. Okay, so maybe W to X on the printout that you've made. I don't know how big it's going to be, but shall we say that it's, I don't know, 22 millimeters long, okay, on your piece of paper in front of you. Okay, so that therefore is the image. That's the image that you've printed out. Now, what if you were told that in actual fact the diameter of the nucleus was 3 microns, okay? So the actual size, excuse me, actual size equals 3 microns. The question would be, what is the magnification of this image? Go ahead, pause the video, work it out, and you can check in a couple seconds. Okay, well, we use this triangle here. So if we want to work out magnification, we do M equals the image divided by the actual. So M equals I over A. So image is 22 millimeters. Actual is 3 micrometers. Now, hopefully, the eagle-eyed amongst you have spotted that these units are not the same. So 22 millimeters, there should be there, and micrometers, it should be there. So they're not the same unit. So we need to make them the same unit. So let's work in micrometers. So how many micrometers would be 22 millimeters? Well, it would be 22,000. So I'm getting a bit pushed to space here, but 22,000 divided by 3, and that equals, calculator here, 22,000 divided by 3 equals around about 7,300 or so. So let's say 7,300 times magnification. Okay, that's magnification. Now I'm going to come on to the final thing. Have I got enough space to do it? I hope so. And we're going to be looking at this diagram right here. Okay, so this final diagram represents a sort of timeline in the stages of a production of a protein. And this is happening in a eukaryotic cell. And we know that because we've got a nucleus. So first of all, let's just sort of label some key features that we've got. So here's the nuclear envelope. This is the nucleus. And this here is the DNA. That's this line there. Now the red bit represents a gene. So that is a gene for a certain protein. So first of all, what happens is the gene here is transcribed. Now we'll learn more about that in the next video. We'll recap it, but you remember that RNA polymerase is the enzyme that transcribes the DNA code into RNA. So this is now messenger RNA. The messenger RNA then exits the nucleus via the nuclear pore, and you can be adding extra details down here if you'd like to, but I'm just going to talk you through it. It exits the nucleus via this nuclear pore here, and now it's the messenger RNA floating in the cytoplasm outside the nucleus. You can see here, if we zoom in a little, you can see here what's going on is that the messenger RNA is actually binding. You can just about see a little purple dot there. It's actually binding to the ribosome. So mRNA binds to the ribosome, and that is where translation occurs. So translation is the code carried on the messenger RNA being turned into a sequence of amino acids, or which is then called a protein or a polypeptide. So here is the protein that is produced. So let's just try and label that. I might just switch up the colors. And I'm just going to label that. That little red thing in there, that is a protein. That is a new protein that has been made. And at the moment, this protein is floating inside the cisternae of the rough ER. So this ribosome is on the rough ER. So this protein is in the cisternae, in this kind of membrane-bound compartment. It's then sent off. So you can imagine that this protein would sort of move down here, down this way, down there, and then it would kind of be stuck in this sort of dead end. And this dead end would sort of pinch off. It would kind of close like this and form a transport vesicle. So this is a vesicle here, and it's on its way to the Golgi. So the protein has been packaged and it lands over here at the Golgi. It'd land over here at the cis face of the Golgi. And it would move through the Golgi. So as it's in the Golgi, it would be modified in the Golgi. I'm going to call this a transport vesicle actually, transport. Modified in the Golgi, and remember we can talk about it might be carbohydrates that are added. It might be cut into a slightly smaller version of the protein, might have an extra bit sort of chopped off. Carbohydrates kind of chopped down. It might be folded in a certain way, or also might have lipids added to it potentially. And once it's been fully processed, it's then going to go into another type of vesicle. And this number 7 here, this one is called a secretory vesicle. So that means that now this vesicle is targeted for, it's heading for the cell surface membrane. Here it's reaching the cell surface membrane at 8, and it's going to fuses with the cell surface membrane, or you could call it the plasma membrane. And then finally it is released. So the protein at 9 is released outside the cell. So this sort of thing, the stages in production and export of a protein, is more complicated in a eukaryote than it would be in a prokaryote. In a prokaryote, something similar would happen with RNA and ribosomes and translation, but it's a lot more complicated in eukaryotes. Right, let's just take a step to zoom out. Okay, there we are. So we've made it to the end of the mind map. So let's take a little sec to reflect on everything we've done. This has covered the whole first sort of chapter of the textbook to do with cells. We've looked at the scales of different things that we can look at, the sizes. We've looked at eukaryotic cells, both plant and animal. We've looked at the major organelles and how they work together. We've also looked at prokaryotic cells, some electron microscope drawings and how to recognize certain structures, and we've briefly touched on how to do calculations relating to magnification. So that's the end of this, which should have taken you around about 2 lessons of work. Please send me a picture of your completed mind map on Teams, and the next task I'm going to be sending you is some multiple choice questions based on all this stuff. Okay, thanks very much. Bye-bye.